up, biology students? Mr. Holloway here. In today's video, we're going to explore photosynthesis a little further. Photosynthesis is an incredibly complex system of reactions and interactions, but in my opinion, it is also a beautifully elegant example of what life is all about. In plants, photosynthesis occurs in the chloroplast, an organelle that has its origins in a mutually beneficial symbiotic relationship founded some 2 billion plus years ago. In this diagram, we can see the genetic material of the chloroplast that supports this theory, but we can also see the important structures and spaces in which photosynthesis occurs, namely the thylakoids and the stroma. Photosynthesis occurs in two stages, the light-dependent reactions and the light-independent reactions, which are also often referred to as the Calvin cycle. During the light-dependent reaction, light is absorbed by pigments in the thylakoid. Water molecules enter during the light-dependent reactions, and this is also when oxygen gas is produced. A couple of important intermediate molecules are produced as well, including a small amount of ATP and a molecule called NADPH, which is like ATP in that it carries a form of energy that can be used later on in photosynthesis. The light-independent reactions occur in the stroma of the chloroplast and do not require sunlight. This part of photosynthesis is powered by the ATP and NADPH produced during the light-dependent reactions. The energy is used to convert carbon dioxide into organic carbon, i.e. sugars. ADP and NADP+, the low-energy forms of ATP and NADPH respectively, are cycled back to the beginning where they can be charged up again with more sunlight. Let's take a closer look at what's happening during the light-dependent reactions of photosynthesis. We're looking at a close-up diagram of a single thylakoid in the chloroplast. The thylakoids are membrane-bound structures, and just like the membranes that surround the cell, this membrane is composed of a lipid bilayer with a variety of proteins and other molecules embedded along with the lipids. Pigments like chlorophyll are also embedded in this membrane, and these pigments absorb light energy in order to begin the process of photosynthesis. Photosynthesis begins in photosystem II, a protein complex embedded in the thylakoid membrane. It is called photosystem II simply because it was discovered and described after photosystem I, which we'll come back to later in the process. Pigments surrounding photosystem II absorb sunlight and transfer this energy to the photosystem II protein complex. This energy is used to catalyze a reaction in which water molecules are split apart, yielding oxygen gas, hydrogen ions, and free electrons. Oxygen diffuses out of the chloroplast and leaves the system entirely at this point. The free electrons follow this path, indicated by the yellow arrow. These electrons are very energetic and have very high energy. Electricity, after all, is the movement of electrons through a conductive material like a wire, and we use electricity to power a lot of things in our everyday lives, so you can think of these high-energy electrons as a kind of electric current running through the proteins embedded in the thylakoid membrane. The energy of these moving electrons is used in a series of reactions that cause additional hydrogen ions to be moved into the interior of the thylakoid membrane. Since these hydrogen ions are moving from an area of low concentration to an area of high concentration, we can think of this as a kind of active transport, powered, in this case, by the movement of high-energy electrons through this series of proteins that we call the electron transport chain. Our electrons lose energy as they move through the electron transport chain, because that energy was used to move hydrogen ions into the thylakoid. When these electrons reach the photosystem I protein complex, which is also surrounded by pigments that absorb sunlight, they are recharged and used in one final reaction, one which transforms low-energy NADP+ molecules into high-energy NADPH molecules, and which is catalyzed by an enzyme called NADP+ reductase. These NADPH molecules will be used later during the Calvin cycle. On the inside of the thylakoid membrane, the area we call the thylakoid space, a high concentration of hydrogen ions has been building up. When water molecules were split at the very beginning of this process, hydrogen ions were produced and released into the thylakoid space. And while these high-energy electrons moved through the electron transport chain, additional hydrogen ions were transported into the thylakoid space as well. As a result, there is a much lower concentration of hydrogen ions in the stroma than there is in the thylakoid. This concentration gradient has high potential energy, like water held behind a dam. If given a pathway, that water will run downhill, and this potential energy will be transformed into kinetic energy in much the same way. As we learned earlier in the year, particles tend to diffuse from an area of high concentration to areas of low concentration, and that is exactly what is happening in this part of the figure. This is called chemiosmosis because, like regular osmosis, which is the diffusion of water from an area of high concentration to an area of low concentration, hydrogen ions produced by splitting water molecules are diffusing from an area of high concentration to an area of low concentration. In this particular case, hydrogen ions diffuse out of the thylakoid space through a very important enzyme complex called ATP synthase. As hydrogen ions move through this enzyme complex, they cause it to spin, much like water running over a water wheel, or through a hydroelectric turbine. This spinning helps ATP synthase to catalyze a reaction that transforms low-energy ADP into high-energy ATP. Like the NADPH molecules produced earlier, these ATP molecules will be used later on to power the Calvin cycle. At this point in photosynthesis, water molecules have been split using the sun's energy, and oxygen gas has been produced. This chloroplast also produced a few energy-carrying molecules, ATP and NADPH. These energy-carrying molecules are used to convert inorganic carbon, i.e. carbon dioxide, into organic carbon in the form of simple sugars like glucose. This happens during the Calvin cycle, which is a cycle in the sense that certain carbon compounds found in the stroma, which are involved in the reactions that eventually produce glucose molecules, are continually reused or recycled by this process. These reactions are energy-absorbing, or endothermic reactions, which take energy from ATP and NADPH to gradually construct molecules with higher and higher energy. In the next few slides, you're going to hear a lot of molecular names and abbreviations. It's not really important to me that you remember all of these names, but it is important to remember that this process ultimately transforms inorganic carbon into an organic, energy-rich, carbon-based molecule called glucose, and this molecule can be used to feed and provide energy to every living organism in the food chain. The first step in the Calvin cycle is called carbon fixation. During carbon fixation, inorganic carbon dioxide reacts with an organic, carbon-based molecule called RUBP. This reaction is catalyzed by an enzyme called rubisco. The organic carbon molecule produced is labeled here as PGA, and each molecule of PGA contains 3 carbon atoms. Energy from ATP and NADPH is used to convert these PGA molecules into a different organic molecule called G3P, which has higher potential energy than PGA. Much of this G3P is recycled, and more ATP is used to turn this G3P back into RUBP, which can react with carbon dioxide to begin the cycle over again. The high-energy ATP and NADPH used during this cycle are transformed into low-energy ADP and NADP+. These low-energy molecules are also recycled and are sent back to be charged up again during the light-dependent reactions that occur in the thylakoid. Meanwhile, a few G3P molecules are removed from the system. The 3 in this G3P refers to the 3 carbon atoms that make up the main structure of this molecule. 2 molecules of G3P, each containing 3 carbon atoms, are combined to make glucose, which contains 6 carbon atoms and has the chemical formula C6H12O6. It takes 6 water molecules and 6 molecules of carbon dioxide to produce 1 molecule of glucose. Part of this reaction requires an input of light energy in order to produce some ATP and NADPH, energy-carrying molecules that are used later to convert inorganic carbon into organic carbon. This process also produces the oxygen gas that we breathe. Obviously, there is more to this important process than we can see here in this simple chemical equation. It is a beautiful and elegant example of biochemistry, a series of chemical reactions involving numerous intermediate molecules that get cycled and recycled over and over again, as well as a great many other different proteins and enzymes that catalyze and facilitate certain aspects of this process. We call this process photosynthesis, and is one of the most important chemical reactions there is. Our knowledge of photosynthesis may one day allow us to engineer the means to produce other carbon-based molecules that might be used to replace fuel sources like gasoline. In the meantime, we benefit from this reaction every single day of our lives as we breathe and eat the products of this chemical reaction in order to survive. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need until you feel like you understand the details of this important chemical reaction that we call photosynthesis.